presentation provides an overview of the methods and results of the Sriba Aquatic Ecosystem Study. To read the study report, please visit dpause.nt.gov.au/Sriba.
Studies a regional survey of the composition of the aquatic biota, including fish, turtles, water plants, and selected aquatic macroinvertebrate taxa to inform decision-making and regulation for the onshore gas industry in the NT. It's one of two projects on aquatic biodiversity in the study area. This project described the composition of the aquatic fauna of surface waters. The other project described the composition of the subterranean fauna present in groundwater, and which won't be covered by this presentation. Study area is an 80,000 square kilometre envelope surrounding areas of potential onshore gas development in the NT. It spans parts of the Daly, Roper and Victoria Wheezo drainage basins, as well as the headwaters of some Gulf draining rivers. Climatically, the study area lies in a transition between the humid subtropical zone and the semi-arid zone, has a wet, dry climate with a marked north-south gradient in rainfall. Average annual rainfall declines from 842 millimetres at Mataranka in the north to 585 millimetres at Elliot in the south. Land tenure is mostly pastoral leasehold land and Aboriginal land with some areas for conservation. There are two sites of conservation significance in the study area. In the north, Mataranka Thermal Pools encompasses most of the area of Elsie National Park, is listed as a wetland of national significance and is listed on the register of the National Estate. In the south, Lake Woods is a large ephemeral wetland that fills after major flooding in Newcastle Creek. It also is listed as a wetland of national significance and can feature large aggregations of breeding water birds in boom years. Study aimed to sample representative aquatic sites across drainage basins and across the range of available aquatic habitats. There is a diverse range of aquatic habitats in the study area, from temporary wetlands, large riverine channel billabongs, rocky gorges and spring-fed perennial rivers. Many natural water bodies have been modified to provide year-round drinking water for cattle, leading to an increase in the number of small permanent water bodies. There were 5 separate components in the aquatic ecosystem study. First was a desktop study using satellite-derived data to map primary aquatic habitat systems, to identify potential refugial habitats and to assist with site selection. The second was a set of investigations led by Dr. Michael Hammer from the NT Museum to resolve the taxonomy of several problematic groups of fish and decapods to provide a better understanding of the species present in the area. The third was a set of field surveys to document the composition of aquatic biodiversity. It was comprised of standardised surveys, a frog survey, and the collection of incidental records of aquatic plants at aquatic survey and frog survey sites. The fourth component was a preliminary study using eDNA to detect significant aquatic species, and the fifth, a synthesis of data to assess conservation values of sites across the study area. Aquatic habitat mapping was based on a 30-year dataset of satellite observations of surface water in the dry season and used a decision rule framework to identify the primary habitat systems. It mapped palestrine, lacustrine, riverine and floodplain systems and provided estimates of the area of each system. The classification was based on the frequency of detection of surface water in individual pixels. For example, frequency values between 0.01 and 0.04 indicate areas of flooding, whilst values between 0.8 and 1 indicate locations more likely to contain permanent water. The mapping results were compared to existing products and found to identify 93% of water features found in the geofabric. The mapping detected 89 potential refugial sites, which may be candidate sites for future monitoring. The second component of the aquatic ecosystem study was a set of genetic investigations to clarify the taxonomy and distribution of several groups of species. The state of knowledge of the taxonomy and distribution of the aquatic biota in the NT is not perfect, and there's still many parts of the NT under-surveyed for aquatic biota. The NT Museum has been conducting molecular analyses on a range of different groups, including purple-spotted gudgeons, fork-tailed catfish, eel-tailed catfish and swamp eels, as part of ongoing collaborative studies on these groups. I'll present here 2 examples from this work. Firstly, those studies found that there are 5 genetic lineages of purple-spotted gudgeons in the NT, 3 of which occur in the study area. So there are potentially 3 species involved, all within different drainages within the study area. This tells something of the story about the state of knowledge of the aquatic biota in the NT. Maganda is a common species across the top end, but there are next to no records in the study area. But what records there are suggest there are possibly 3 species present. Another example is from recent work by the museum on red-clawed Cherax quadricarinatus using specimens provided by the Schreiber work. There are 2 known genetic lineages, a northern and a southern lineage, but recent analyses turned up a 3rd lineage of Cherax quadricarinatus, which is so far unique to the Little Roper River near Mataranka. And we can say that this area is significant for the genetic diversity of this group. We lodged voucher specimens with the NT Museum, so they are available for reference and future taxonomic study. There's no shortage of questions on the taxonomy of freshwater invertebrates in Northern Australia. So another example of freshwater crabs, they're common and widespread. They turn up nearly everywhere in the drier country. There are different colour forms, but we have no idea if they represent one or several species. They're clearly a priority taxa for genetic research. Genetic analyses should be a routine part of the toolkit for freshwater biodiversity assessment. Third component are field studies to document the composition of aquatic biodiversity. We did a comprehensive survey of the aquatic biota at 44 sites. These sites were distributed across the study area, but with a northern bias. Sampling at each site was quite intensive. At most sites, we spent about 18 hours on site. And at most sites deployed a standard set of 8 sampling methods, including baited traps, pike nets, gill netting, backpack electrofishing, cast netting, turtle traps, spotlight searches, and benthic sampling of littoral habitats. Sampling spanned 2 dry seasons, with each site visited on only 1 occasion, so there's no capacity to describe seasonal variation at the level of individual sites. We used multiple sampling methods for fish to cover different habitats and fish behaviours. No one method would have adequately sampled the entire range of species present. Pike nets target small active fish in shallow littoral areas. Electrofishing targets small cryptic fish in littoral areas, particularly with complex habitat like snags. And gill nets target larger active fish in deeper waters. For turtles, we found that not all turtle species enter traps. And for some species, we had to rely on spotlighting and dip netting. For benthic macroinvertebrates, we used standard pond net to sweep net along 10-metre transects. From the standardised sampling at 44 sites, we found 291 species-level taxa, not all named species, and some were unnamed morphospecies. We used specialist taxonomists where possible for most groups, including fish, aquatic beetles, molluscs, and antiids. We found 36 species of fish, 11 species of aquatic or semi-aquatic reptiles, including 7 turtle species. About 84% of the total number of aquatic species are invertebrates, with aquatic beetles comprising the largest single group of more than a quarter of all species. The top end is a diverse aquatic beetle fauna, and it's not surprising to find that beetles are well represented. There are more species present in one family of beetles than there are species of fish. We found 36 species of fish from 19 families. There's a set of near ubiquitous species, including spangled grunter, checkered rainbow fish, and northwest glassfish found at most sites at which fish are present. Spangled grunter is especially good at occupying temporary habitats. If there's one species of fish present at a site, it will invariably be spangled grunter. At the tail of the distribution, there are 8 species only recorded at 1 or 2 sites, including freshwater sole and freshwater garfish. Most species of fish were recorded at sites on rivers along the northern margin of the study area. All but 3 of 36 species were recorded at sites in the Upper Roper. The 3 that weren't include silver catfish, Orochalus argenteus, which has a distribution extending into arid parts of Queensland, the giant glassfish Parambassus gulliveri, which has a seemingly odd distribution with a gap between western and eastern populations, and the western rainbow fish, which in the study area is found in the Daly but not in the Roper. In contrast, the entire Newcastle Creek system has a total of only 6 species. There were 7 species in total, each different in details of habitat and ecology. The Worrell's turtle is the common species in the area and was present at 18 of 44 sites. Cairns turtle appears to be an arid country specialist occupying temporary water bodies. One of the common names I've read for this species is the stinky armpit turtle because it exudes a defensive pungent yellow fluid from the groin and armpit areas. The species of most conservation interest is the Gulf snapping turtle, Elsea lavricorum, which is found at 3 sites in the Upper Roper. There is a diverse range of aquatic beetles present in the study area. It includes a few near ubiquitous species, but also a large number of species which were only rarely recorded. And most of these species appear to be widely distributed across northern Australia. This is a stacked bar plot showing variation in composition of taxa groups at each of the 44 sites, arranged in a clockwise fashion from the south. The inland Newcastle Creek system sites on the left-hand side of the plot and the Upper Roper River sites on the middle right-hand side. And there are regional differences in total species richness, in the richness of fish, shaded in grey, in the richness of aquatic reptiles, shaded orange, and the richness of molluscs, shaded brown. We conducted a multivariate analysis of species composition to see how species are sorted amongst sites. Classification of 291 species identified 12 site groups, including 9 multi-site groups and 3 groups comprising a single site. Groups form 4 high-level clusters corresponding to spring-fed sites in the Upper Roper and Roper tributaries, non-spring-fed sites of the Daly and Roper tributary streams, inland sites mostly on Newcastle Creek and Western Creek, and lastly, a miscellaneous group of sites on wetlands, some sampled from remnant water bodies after the wetland is dry. I've highlighted here Group 4, which includes 4 sites on riverine channels in Newcastle Creek, which features high turbidity and low conductivity. Group 7 are mostly spring-fed sites on the Upper Roper, with high conductivity and low nutrients, chlorophyll and turbidity. And Group 9 is a spring-fed sandstone stream. It's the only example of this type of habitat in the data. We collected data on water quality at all sampling sites to provide baseline data and data for interpretation of survey data. Bar plots showed values for 6 water quality variables ordered by latitude. Sites located in the southern portion of the study area have high values of turbidity, total nutrients and chlorophyll, and low values of conductivity. Sites in the Upper Roper in the northern portion feature low values of turbidity, total nutrient and chlorophyll, and high values of conductivity. Same result is shown in bubble plots with water quality variables or values overlain on an ordination of species composition. Group 4 sites on Newcastle Creek feature high chlorophyll and low conductivity, with the reverse for Group 7 sites in the Upper Roper. We surveyed frogs at an additional set of sites using passive acoustic monitoring. We deployed digital recorders at 31 sites in February 2022. Recorders were scheduled to record 1-minute files every 10 minutes, resulting in over 12,000 recordings, summing to nearly 9 days of continuous listening. In the future, it is likely this volume of data will be analysed using machine learning to recognise species. But in the absence of that, we manually identified species by visual inspection of spectrograms of 700 recordings during peak calling times. This is a demonstration of the core signatures of different species that are viewable on a spectrogram. There's a set of everywhere species which occur at most sites, including Cyclorhina australis, Litoria pallida, Litoria rothii, and Litoria rubella. And we identified 17 species from recordings and found an additional 8 species during ground surveys. We surveyed aquatic plants at each site by a non-systematic collection of specimens which were subsequently identified by staff at the endoherbarium. We identified 84 species. The only species of conservation note was Dentella minutissima, which grows on dry and clay fringe of water bodies and cannot be considered a fully aquatic species. We conducted targeted surveys of 2 of the 4 significant species known in the study area. They're classified as significant by virtue of the fact that they are listed under either territory or national environmental legislation. Under the Commonwealth EPBC Act, the Gulf snapping turtle is listed as endangered, and the large-toothed sawfish is listed as vulnerable. Both are quite cryptic species that can be difficult to detect, but particularly the Gulf snapping turtle. We failed to catch any Gulf snapping turtles in any traps, but found them at 3 sites by spotlighting along the edges at night from a boat, which is fine with waters of high visibility and in areas with low crocodile risk. But for both species, there's a need for novel, non-invasive survey methods such as environmental DNA, which detect traces of the animal's DNA in water samples. The method has been used previously with success to detect sawfish and other species of Alcea. We collected water samples from 15 sites in the Roper for detection of sawfish and turtle DNA by the eDNA labs of James Cook University. We found no sawfish DNA. We found Gulf snapping turtle DNA at 6 of 15 sites. But these are preliminary results, and we need to be sure that we are reliably differentiating between the two species of Elsea that may be present. We are looking at different ways of presenting information on the conservation value of sites. In the first, total species richness, which is the sum of all species detected during routine surveys at a site. We found here that high values occur at sites along the northern margin of the study area, but especially at sites within the upper Roper. There's another way of looking at the patterns of diversity. This is richness data per group standardised by the maximum number of species observed at a site for that group, and then averaged across groups to give mean standardised richness. And it demonstrates a similar pattern as previously. The highest values occur along the northern margin of the study area and in the upper Roper sites. This is the number of rare taxa recorded per site, or significant taxa. Of the 4 listed species, 3 species were recorded at sites in the upper Roper, with a 4th probably present based on previous records, but was undetected during our study. And lastly, the number of unique taxa. That is the number of species recorded at only a single site. Now, there were 60 species recorded at or found at a single site. This metric is influenced by the representation of different types of sites. So we have found a similar pattern as previously, with sites in the upper Roper standing out. Perhaps a better way to approach this is to look across the span of available evidence from different sources. The upper Roper sites have distinctive water quality from groundwater inflows with low chlorophyll, low nutrients, allowing high light penetration for the growth of submerged macrophytes. There is the presence of significant species and evidence of unique genetic lineages based on crayfish data. There's high species richness in surface waters with 33 species of the 36 species of fish found in the area. There's high stygofauna richness with 26 of 28 known species present in the vicinity. These all point to the upper Roper regions having high conservation values which were previously recognised by its listing as an NT site of conservation significance called Mataranka thermal pools. The last slide but one summarises the main results of the study and lists some knowledge gaps. Firstly, the study mapped the primary aquatic habitat systems in the study area. We conducted genetic investigations to identify the ecological assets in the study area. We documented species-level composition of the aquatic biota in the study area. We identified 291 aquatic species from standardised surveys, as well as 25 frog species and 84 aquatic plant species, totalling 400 species. The probable total aquatic richness would be in excess of 500 species. We confirmed that biodiversity and conservation values are centred in the upper Roper River. However, we acknowledge that water bodies on intermittent systems elsewhere in the region may have high conservation value as waterbird habitat. We trial eDNA methods for the detection of significant species. And lastly, 3 knowledge gaps. Firstly, we require information on seasonal variability in aquatic faunal assemblages. We require better knowledge on the ecology of significant species, including Gulf snapping turtle. And lastly, and not so much a knowledge gap as an information need, the development of eDNA-based methods for routine biological monitoring and for the detection of significant species. I'd like to acknowledge the support and assistance received from members of the survey team, from researchers, ranger staff, station managers, and particularly acknowledge the generous assistance from traditional owners and Indigenous rangers in allowing access to Aboriginal land. Further information on Sriba projects can be found at this website.